दिल्ली में मिल रहा। पॉपकॉर्न फेंक दें।
चलो हम अब ऑनलाइन जा रहे हैं, अब बात नहीं कर पाऊंगा, फेंक दो। So, a very good afternoon to one and all present. Today we will be discussing the questions which are related to week 3 session. So, this week 3 is basically related to the differential gene expression and how the differential gene expression works and the basics about transcription and translation. So, all these questions related to all these topics will be dealt in this class. So, I have already informed in my previous class that I won't be able to take the class on Tuesday. That's why we are having class today. On Sunday, I hope you have all got the information and the mails regarding it. So, first of all, we need to know the basics of differential gene expression. So, differential gene expression, just a quick recap of what differential gene expression means that we have also discussed in our previous lecture. So, differential gene expression means that all the genes are present in each and every cell of the body, but all the genes are not expressed in each and every cell of the body because of the property of differential gene expression. That means despite of the fact that all the genes are present in each and every cell of the body, their expression is spatio-temporally controlled. In a cell and state specific manner, their expression is controlled. What does that mean? It means that some genes will be expressed only at certain developmental stage of our life, and they will not be developed in other or later stages of life. Likewise, some genes will be expressed in particular organs or cells, and they will not be expressed in other organs and cells. For example, eye gene, which is responsible for eye color, it is present in your liver, heart cells also, but those genes are not expressed in your liver or heart cells. So those genes are differentially or specifically expressed only in the eyes. So this is what we call as differential gene expression. That means the expression of gene is cell type and state specific. Okay, so how does it happens? It happens because of various phenomena that happens or various biological processes that happens inside the body. For example, alternative splicing or alternative taking of the poly A binding sites and many more. So some phenomena actually contribute to the differential gene expression and ultimately leads to variability in the expression of genes in each and every cell of the body. Okay, so this is like a basic gist of what differential gene expression is. Now let us move to the first question. So the first question is, which of the following may result from mutation in cis regulatory elements? Okay. The question is, key if there is a mutation in the cis regulatory elements, which all can be the consequences of that? Okay, so the options are change in localization of mRNAs, second, inability to bind splicing enhancers, inability to bind RNA polymerase, misregulation in splicing, synthesis of truncated proteins, or inability to bind the transcription factors. Okay, this is the question. I'll just mark it as A, B, C, D, E, and F. Okay, so options are change in localization of mRNAs, inability to bind to splicing enhancers, inability to bind to RNA polymerase, misregulation in splicing, synthesis of truncated proteins, and inability to bind to the transcription factors. These are the uh, just a minute. Just want to. So I'll give you 2 minutes, and after 2 minutes, I'll switch the screen and then we'll discuss the question. Okay. Think of the answer. Okay, so now I am the screen. Switching the screen now. Okay, so the question was, which of the following may result from mutation in cis regulatory elements? And the options were change in localization of mRNAs, inability to bind to the splicing enhancers, inability to bind to the RNA polymerase, misregulation in splicing, synthesis of truncated proteins, and inability to bind to the transcription factor. And the correct answer is change in localization of mRNAs, inability to bind to RNA polymerase, misregulation in splicing, synthesis of truncated proteins, and inability to bind to the transcription factors. So, Now let us try to see each and every option. So the first is change in the localization of mRNA. Is this a correct statement or not? Yes, it is a correct statement. Why it is a correct statement? Because cis regulatory elements in the 3' UTR regulate the mRNA localization in the cytoplasm. So first of all, before answering this question, let us try to understand what is the meaning of cis regulatory elements. So cis regulatory elements are composed of DNA. Basically, they are the non-coding regions containing binding sites for transcription factors and other regulatory molecules that are needed to activate or sustain transcription. Promoters and enhancers are the best understood type of cis regulatory elements. So, cis regulatory elements means these are the DNA sequences which are present in cis manner with the uh, regulatory part. For example, these are your promoters and enhancers. These are your cis regulatory elements. So this first point was change in the localization of mRNA. So these cis regulatory elements in the 3' UTR, they regulate the mRNA localization in the cytoplasm, and therefore their mutations can mislocalize the transcript. So this is the correct statement. Second is inability to bind to the splicing enhancers. This is not a correct statement. Okay. Then inability to bind to RNA polymerase, yes, because if there is a mutation in the core promoter, um, it can disrupt the cis regulatory RNA polymerase binding. Okay, so therefore this is also a correct answer. Misregulation in splicing, yes, it is again correct. Why? Because cis regulatory elements at exon intron boundaries or intronic splicing enhancers are required. So if there is a mutation, it causes exon skipping, or it can lead to mis-splicing also. So what is there? What they are saying, that these cis regulatory elements, they are present in the intron exon boundaries. Okay, so they are present in the intron exon boundaries. And if there is a mutation, it can cause exon skipping also, or mis-splicing also. Therefore this is again the correct answer, option. Then synthesis of truncated proteins, again it is correct. Why it is correct? Because if splicing is disrupted, then obviously the protein that is formed will also be disrupted. This is so but obvious, है ना? So if splicing is disrupted, uh, mm, just a minute. So if splicing is disrupted, then obviously the protein which is getting formed will also be disrupted. So yes, synthesis of truncated protein is the correct one. Then inability to bind to the transcription factors, obviously, because these regulatory elements are the DNA sequence where the transcription factors come and bind. So if there is a mutation in the cis regulatory elements, obviously the ability of the transcription factor to bind would be compromised. So this is also uh, correct statement. So transcription factors are trans-regulatory elements because they are coming from outside and they are binding. And cis-regulatory elements are promoters and enhancers which are present in the DNA sequence. Okay. Now the next question is, which of the following statements regarding gene expression are true? A paternal imprint allows expression from the paternal allele. Let me just mark it as A, B, C, D and E. Okay. So A is a paternal imprint allows expression from the paternal allele. B, alternative 5' and 3' splicing are mutually exclusive. C, alternative splicing is the main mechanism by which protein isoforms are generated. D, protein isoforms may have opposite effects, and E, a mutation in the 5' splice site prevents the binding of U1 snRNA. Okay, so you have to tell that which of the statements regarding these are true. So try to answer this question. After 2 minutes, I'll switch the screen. Think. After 2 minutes, I will switch the screen and then we will discuss it, okay? One more minute left. Thirty seconds more. Okay, so now I am switching the screen. Hmm. Okay, so the question is, which of the following statements regarding gene expression are true? Okay, so the correct answer is alternative splicing is the main mechanism by which protein isoforms are generated. Protein isoform may have opposing effects, and a mutation in the 5' splice site prevents binding of U1 snRNA. Now, let us try to understand this question. Okay, so the first one is a paternal imprint allows the expression from the paternal allele. Is it true? No, because imprinting means silencing or masking of one of the allele. Okay, so if the it suppose imprinting means that if the gene is if a gene see a gene has two allele, one comes from father and the other comes from mother. If the paternal allele is imprinted, that means paternal allele is silenced, it is masked. So the expression will come from the maternal allele, because imprinting means silencing of imprinting means silencing. Imprinting means silencing. So if the paternal allele is imprinted, then the expression will be from the paternal allele? No, the expression will be from maternal allele in that case, because the paternal allele is silenced. So this is wrong statement. Second is alternative 5 prime and 3 prime splicing are mutually exclusive. Are they exclusive? No, because they are different modes of splicing, but they are not mutually exclusive. That means they can happen within the same gene. Okay, it can happen within the same gene. So are they mutually exclusive event? No. So this is again wrong. Then alternative splicing is the main mechanism by which protein isoforms are generated. Yes, it is true. It is the dominant mechanism in eukaryotes for generation of protein isoforms. Then protein isoforms may have opposing effects. True, it can have opposing effects because it depends on what kind of genes are getting spliced and what kind of isoform is formed. For example, uh, pro-apoptotic versus anti-apoptotic form of BCLX. So BCLX is a gene, and it can lead to pro-apoptotic isoforms and anti-apoptotic. Both are completely opposing. Both have completely opposing effects. So protein isoforms may have opposing effects. True. Then a mutation in the 5 prime splice site prevents the binding of U1 snRNA. Exactly. Why? Because the 5 prime splice site is recognized by the U1 snRNA. So if there is a mutation in the 5 prime splice site, obviously the binding of U1 snRNA will be affected. So this U1 snRNA, it recognizes the 5 prime splice site. 5 prime splice site. So again, this is a true statement. So your accepted answers would be alternative splicing is the main mechanism by which protein isoforms are generated. Protein isoforms may have opposite effects, and a mutation in the 5 prime splice site prevents the binding of U1 snRNA. Okay. Now the next question is, select the correct statements regarding the regulation of mRNA. This is a question about the mRNA regulation. Okay, the options are, again, I'll just mark it as A, B, C, D, and E. Okay. Um, 5 prime methylation of A is 5 prime methylation of unmethylated mRNA aids in achieving translational competency. B. Poly A tail length affects mRNA stability. C. 3 prime UTRs are not required for mRNA stability but are required for translational regulation. D. Cis acting proteins bind to splice sites and promote splicing. And E. Masking loops the mRNA by binding to proteins bound to the 5 prime and 3 prime ends of the mRNA and prevents translation. These are the options. Okay, so think and then we will discuss after 2 minutes. I'll switch the screen. Meanwhile, you try to answer the question. One more minute. Think and try to answer the question. Okay, so I'm switching the screen now. Hmm. So the correct options are 5' methylation of unmethylated mRNAs aid in achieving translation competency. Poly A tail length affect mRNA stability. Masking loops the mRNA by binding to proteins bound to the 5' end, 3' ends of the mRNA, and prevents translation. These are the correct options. Now let us try to understand uh, each and every point. So the first is 5' methylation of unmethylated mRNAs aids in achieving translational competency. Is it true or not? It is true statement. Why? Because the 5' cap is essential for translation initiation and protection from degradation. 5' methylation of unmethylated mRNA. What is the meaning of 5' methylation of unmethylated mRNA? It means 5' capping. This means 5' methylation of unmethylated mRNA is nothing but 5' capping. So does it helps in translation? Obviously, because the 5' end of the mRNA, because of 5' capping, is protected. Otherwise, mRNA can be degraded very easily because of the exonucleases which are present. Also, this mRNA, when it goes to the cytoplasm for translation, okay, mRNA goes to the cytoplasm for translation. And cytoplasm has what kind of environment? Cytoplasm has a reducing environment. That means that the mRNA can be easily degraded when it comes to cytoplasm. Therefore, before the mRNA is released into the cytoplasm, it is very much protected by various mechanisms, by various ways. And one of the mechanisms or one of the ways by which the mRNA is protected is through 5' capping. So there is a cap at the 5' end of the mRNA which is protecting it from the attack of exonucleases and protecting it from degradation. So this 5' methylation of unmethylated mRNA is nothing but 5' capping. So this is correct. Second is poly A tail length affects mRNA stability. Again, it is a correct option because longer the poly A tail, greater would be the mRNA stability and greater would be the translational efficiency. Shortening leads to degradation. So again, this is also correct. Then 3' UTRs are not required for mRNA stability but are required for translational regulation. See, and if it is wrong because when the mRNA is stable, then only it can undergo translation. Without the stability of mRNA, it cannot undergo a translation. So, and also 3' UTR, it contains elements like AU rich elements, mRNA binding sites, polyadenylation signals that regulates both stability also and it regulates translation also. So this is a wrong statement. Then cis regulatory proteins bind to splice sites and promote splicing. So this is again wrong Because splicing is promoted by trans-acting proteins that bind to cis-acting RNA sequences. So cis-acting proteins bind to splice sites? No. Anything that is getting bind to any kind of sites are always trans-acting elements. So splicing is promoted by trans-acting proteins that bind to cis regulatory RNA sequences. So again, this is a wrong statement. Then masking, it loops the mRNA by binding to proteins bound to the 5' and 3' ends of the mRNA and prevents translation. Yes, it is a correct statement. Why it is a correct statement? Because masking links uh, uh, looping the mRNA and blocking translation until the cytoplasmic polyadenylation occurs. So therefore it is a correct statement. So I hope this question is clear to all of you. Now we'll come to the next question. Next question is, identify the correct statements regarding small RNA mediated regulation of gene expression. Small RNA, small RNA mediated regulation of gene expression. Okay, let me just mark it as A, B, C, D and E. Okay. So option A is complete complementarity of the microRNA with the target sequence enables selectivity. B. MicroRNAs primarily target the 3' UTR of an mRNA. C. RNAi means RNA interference is required for endogenous genes but not for the exogenous genes. D. Arginate protein is responsible for binding to target mRNA and inducing cleavage. And E, the seed sequence complementary to the target site in the 3' end uh, of the miRNA. Okay, so these are your options. Think and try to read, and then we'll discuss after 2 minutes. Think. One more minute left. 30 seconds. Okay. Let us try to answer this question now. The question says identify the correct statements regarding small RNA mediated regulation of gene expression. And the options are complete complementarity of the microRNA with the target sequence enables selectivity. MicroRNAs primarily target the 3' UTR of the mRNA. RNA interference is required for endogenous genes but not for the exogenous genes. And argonate protein is responsible for binding to target mRNA and inducing cleavage. And the seed sequence complementary to the target resides in the 3' end of the mRNA. The correct options are microRNAs primarily target the 3' UTR of an mRNA, and argonate protein is responsible for binding to target mRNA and inducing the cleavage. So let us try to understand the points here. The first one is complete complementarity of the microRNA with the target sequence enables selectivity. It is incorrect. Why? Because in animals, partial complementarity is enough for silencing. See, there can be two things. Either there can be the degradation of the mRNA or there can be the silencing. So for degradation point, complete complementarity is required. But for silencing, only partial complementarity is also fine. So in animals, especially in animals, partial complementarity with the seed sequence is mostly seen. So partial complementarity leads to silencing, and complete complementarity, which is mostly seen in plants, it leads to cleavage. So this is wrong. Then microRNAs primarily target the 3' UTR. Yes, it is correct. So because most of the miRNA and mRNA interaction, it occurs at the 3' UTR, which regulates the stability and translation of that particular protein which is going to be formed from that mRNA. So yes, this is the correct statement. Second is RNA interference is required for endogenous genes but not for the exogenous genes. No, it is an incorrect statement because RNA interference ka jo, the machinery of RNA interference, it regulates both endogenous genes, for example, uh, through miRNAs, and exogenous or foreign genes through siRNA. So RNA interference can be performed through miRNA also and through siRNA also. So with the help of miRNA, endogenous genes are controlled, and the exogenous or the foreign genes are controlled via siRNA. So both endogenous and exogenous genes are controlled. So this is again a wrong statement. We cannot say that RNA interference is required only for the endogenous genes. So remember, miRNA, it regulates endogenous genes, and siRNA, it regulates the exogenous genes. Okay. Next point was argonate protein is responsible for binding to target mRNA and inducing cleavage. Yes, it is correct. Argonate is a part of the risk which bind to the RNAs, and if complementarity is high, argonate cleaves the target mRNA. So this is the correct statement. Next is the seed sequence complementarity to the target resides in the 3' of the mRNA. The seed sequence at the 5' of the mRNA is typically 2 to 8 nucleotide. So this is your again wrong statement. Okay. I hope this is clear. Now let us come to the next question. So there are many. It's a big question. Let me just. Mm, okay. Yeah. Okay, fine. Um, Anyways, let me just mark it as A, B, C, D so that it is easier to point out the options. A, B, C, D, E, and F. Fine. So the question is identify the correct statements regarding the control of gene expression. A. Transcription factors are dispensable in prokaryotes. B. Enhancers increase the rate of transcription by binding to regulatory sequences. C. Histone deacetylase recruited by transcription factors result in reduced transcriptional activity. D. Transcription factors are trans-regulatory factors, but microRNAs are not. E. Transcription elongation complex releases RNA polymerase II from the mediator complex. And F. An enhancer of a gene can bind only to one transcription factor, while the transcription factor may bind to multiple enhancers. Okay, these are some of the statements regarding the gene expression control. Okay, there are one, 6 statements are there. Think carefully and then you try to answer this. Okay, I give you 3 minutes for this. See the options, try to understand each and every option and then try to answer it. Okay, see all the options very carefully. All the options must be read carefully before answering. Let's switch the screen and try to understand the options now. So, the correct statements are: transcription factors are dispensable in prokaryotes. Histone deacetylase recruited by transcription factors result in reduced transcriptional activity. Transcription elongation complex releases RNA polymerase 2 from the mediator complex. Okay, so before trying to understand this question, let us try to understand what is the difference between cis regulatory elements and trans regulatory elements. So cis regulatory elements, they are located on the same DNA molecule. This is important. Cis regulatory elements are located on the same DNA molecule as the gene they regulate, whereas trans-regulatory elements are located on different DNA molecules. This is the main difference between cis-regulatory elements and trans-regulatory elements. Okay, cis-regulatory elements are located on the same DNA molecule as the gene they regulate, whereas trans-regulatory elements are located on different DNA molecules from the gene that they regulate. First thing, and they are typically short DNA sequences that act as binding sites for the transcription factors. They are the binding sites for transcription factors, whereas trans-regulatory elements are the transcription factors because they bind to the cis-regulatory elements. Okay, these genes are referred to as transcription factors or transacting factors because they bind to the cis regulatory elements. They can bind to the cis regulatory elements. So examples of cis regulatory elements are promoter, enhancers, or silencers. For example, a promoter cis regulatory element is responsible for initiating the transcription of a gene by binding transcription factors or RNA polymerase to the gene's promoter region. So this is a difference between cis regulatory elements and trans regulatory elements. So this is important. Cis-regulatory elements are the DNA sequences located near the gene they regulate, while trans-regulatory elements are the genes or proteins that can regulate the expression of target genes located on different DNA molecules. So cis-regulatory elements essentially are located on the same DNA molecules, whereas trans-regulatory elements are located on the different DNA molecules. So basically, trans-regulatory elements are your transcription factors and cis-regulatory elements are generally promoters and enhancers. So these trans-regulatory elements, they come and bind to the cis-regulatory elements. So here the example of trans-regulatory elements is transcription factors. And here promoters, enhancers, silencers. Okay, so I hope the difference between cis regulatory elements and trans regulatory elements is clear to you. Now let us again come to this question. The question was identify the correct statements regarding the control of cis gene expression. And the correct answers are, options are: transcription factors are dispensable in prokaryotes. Histone deacetylase recruited by transcription factors result in reduced transcriptional activity. Transcription elongation complex releases RNA polymerase 2 from the mediator complex. Okay, these are your correct statements. Now, let us try to understand each and every statement of the question one by one to have a proper, clear understanding of the question. Okay, so the first point is transcription factors are dispensable in prokaryotes. Yes, it is true. Why? Because when we talk about we are talking about prokaryotes. So whenever we are talking about prokaryotes, the RNA polymerase holoenzyme, it can initiate transcription without the need of additional transcription factors unlike in eukaryotes where multiple transcription factors are essential. So whenever we're talking about prokaryotes, multiple transcription factors are required. Here, the RNA polymerase can initiate the transcription without the need of the multiple list of transcription factors which are involved in the case of eukaryote. So yes, this is a correct statement. Then next is enhancers. Increases the rate of transcription by binding to the regulatory sequences. No, because enhancers do not bind to the regulatory sequences. Regulatory sequence or the factors, they come and they bind to the enhancer. Okay, so this is your wrong statement. Then histone deacetylase is recruited by transcription factor reduces reduced transcriptional efficiency. No, it is again a wrong statement. Okay. This is a correct statement. Why? Because acetylation increases the transcription efficiency. Okay, acetylation. This increases transcriptional activity. So if deacetylation is happening, then it will reduce the transcription efficiency. So histone deacetylases, it will cause the deacetylation. So if there is deacetylation of the transcription factor, then it will reduce the transcriptional activity. So this is correct. Then transcription factors are trans-regulatory factors, but microRNAs are not. Is this a correct statement? No. This is again an incorrect statement. Okay. Then transcription elongation complex releases RNA polymerase 2 from the mediator complex. Yes, it is correct. I hope this is correct. Yes, this is correct because after initiation, the elongation complex, it helps RNA polymerase 2 to escape from mediator, and then it promote the proximal pausing which enables the productive mRNA synthesis. Okay, so this is wrong. This is correct. An enhancer of a gene can bind to only one transcription factor, while the transcription factor may bind to multiple enhancer. Again, this is a wrong statement. Okay, so I hope all the options are clear to you. See, this last option says an enhancer of a gene can bind to one transcription factor, while the transcription factor can bind to multiple enhancer. Is it correct? No, because a single enhancer can bind to multiple transcription factors in combination. Not one by one also. In combination, it can bind to multiple enhancers. And that's how combinatorial regulation works. So a single enhancer can bind to multiple transcription factors. Important. A single enhancer can bind to multiple transcription factors. I hope this is clear to you. Okay, next question is sorry. Hmm. Ha. This is the next question. A, B, C, D, and E. Identify the correct statements regarding translation and post-translational regulation. A. Mutations in the Cossack sequence will hinder efficient translation. B. Deletion within the Pribnow sequence will affect histone modifications. C. Poly A binding protein binds to the poly A tail and promote translational quiescence. D. miRNAs cannot affect the translation of mRNAs bound to the ribosome. And E. mRNAs localized to specific regions within the embryo can be selectively translated. So these are the statements regarding translation and post translational regulation. Go through the options and then we'll see. Okay, try to answer this question. After 2 minutes I'll switch the screen. Okay, and then we'll discuss the question. You have 2 minutes to answer this question. Okay, 1 more minute. Okay, now I'm switching the screen. So identify the correct statements regarding translation and post translational regulation. The correct statements are mutation in the Kozak sequence will hinder efficient translation, and mRNAs localized to specific regions within the embryos can be selectively translated. Okay. Okay, so let us try to understand the question. Mutations in the first is mutation in the Kozak sequence will hinder translation efficiency. Obviously, it will hinder because the Kozak consensus sequence it surrounds the start codon, which is AUG. So, if there is a mutation, that means the initiation of the translation would be obviously affected because it is the Kozak sequence is present at the start codon only. Then deletion within the Privnov sequence will affect histone modifications. This is again incorrect. Why? Because the Privnov box is a prokaryotic promoter element. It has nothing to do with histones, which are actually absent in the case of prokaryotes. So this is your wrong statement. Then poly A binding protein binds to poly A tail and promote translational quiescent tail and promoter translational quiescence. Again, this is incorrect because poly A binding protein it actually enhances the translation by binding to the poly A tail with the 5' cap complex, and therefore translational quiescence is maintained by protein like masking but not with the help of poly A binding protein, because PABP, this poly A binding protein, it actually promotes or enhances the translation. So this is an incorrect statement. Then miRNAs cannot affect the translation of mRNAs bound by the ribosome. Again, it is incorrect because miRNAs, microRNAs, it can repress the translation even after ribosome binding if they can induce ribosome block. Okay. And they can induce the ribosome block or ribosome elongation. So mRNAs, it can repress translation even after binding of the ribosome. So this is again incorrect statement. Then mRNAs localized to specific regions within the embryo can be selectively translated. Yes, it is correct because, for example, bicoid, nanos mRNA, if you have studied. So localized mRNAs are stored in repressed state and translated only at the right time. So we know that the translation, this expression is cell and developmental state specific. So when I say the expression is cell and developmental state specific, this automatically means that the expression of certain genes, and when I say the expression of certain genes, I'm talking about the ultimately the protein formation, because the ultimate expression of any gene is the formation of protein, because whenever it forms a protein then it shows its effect. So the ultimate test of expression is whether it is forming a fully functional protein or not, and that is cell and developmental state specific. So localized mRNAs, they can be stored in repressed state and they can be translated at right place and right time. Okay, so mRNAs localized to specific regions within the embryo can be selectively translated. So I hope this question is clear. Now we'll come to the next question. It's a numerical type. From here now, all the questions are numerical type questions. Okay, so a plant bearing red flower is crossed with a plant bearing white flower. Assume that both plants chosen here are homozygous and that the red and white colors are dominant and recessive phenotypes respectively. Calculate the percentage of red flowers in the F1 generation. Okay, this is the question. I'll again repeat the question. A plant bearing red flower is crossed with a plant bearing white flower. Okay, assume that both plants chosen here are homozygous and that the red and white colors are dominant and recessive phenotypes respectively. Calculate the percentage of red flowers in the F1 generation. In the F1 generation, you have to calculate the percentage of red flowers, which are the dominant types. Okay, so I'll give you 3 minutes to solve this question, and then I'll switch the screen and then we'll discuss the question. Okay, it's a very easy question, nothing much to do. If you know how to make a Punnett square, how to draw a Punnett square, then it is very easy for you to solve this question. Fine. So try to solve this question. After 2 minutes, we are going to discuss it. Try to understand the question and try to solve this question with the help of Punnett square. With the help of Punnett square only you can solve this. Two more minutes left. One more minute left. Okay, so I'm switching the screen now. A plant bearing red flower is crossed with a plant bearing white flower. Okay. Assume that both plants and plants chosen here are homozygous and that the red and white colors are dominant and recessive phenotypes respectively. Calculate the percentage of red flowers in the F1 generation. Hmm. Okay. So red is dominant and assume that both plants here are homozygous. So is it fine now? Yeah. So red flower, homozygous. So it would be R R. And white flower, homozygous. So it would be R R. This is dominant and this is uh, recessive. So now what will be the gametes? Gametes would be R and R. Now let us make a Punnett square out of it. See, assume that both flowers chosen here are homozygous. So this is homozygous, homozygous, and red flower is dominant and white flowers are recessive. Calculate the percentage of red flowers in the F1 generation. That how many red flowers are formed in the F1 generation. So let us try to do it with the help of the Punnett square. R, 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 and R, R. So all are heterozygous, but all are red. All are red color flowers. So this means there are 100% red flower. And the answer is 100. So is it clear to everybody? It's very easy. You just have to calculate the percentage of red flowers. All are heterozygous, but since red is dominant over the white, therefore all phenotypically all will be red colored flower. Because we have to access only the F1 generation. Now let us come to the next question. A cross between two plants that are heterozygous for seed color. Green is the dominant and yellow is the recessive, and seed coat. Smooth is dominant and rough is recessive. Yielded 900 progenies. Okay. A student collected all the samples with smooth seeds. What percentage of plants bearing smooth seeds will have yellow seeds? Okay. So this question you have to solve. Again, easy question. Two characteristics are given here. You see, seed color is given and seed coat is given. In earlier in this question, only one of the characteristic, which is color of the flower, was there. Red color or white color. So it was a monohybrid cross. It is a monohybrid cross. This is a monohybrid cross because only one character is getting dealt here. But here you can see there are two characteristics. First is seed color and the second one is seed coat. That means two characteristics. So I'm just giving you a hint for you to solve, which is that this is a dihybrid cross. Two characteristics are there, which means it's a dihybrid cross. So you have to make Punnett square related to dihybrid cross, and dihybrid cross comes in the ratio of 9:3:3:1. These are all the hints that I have given you. Now you try to solve this question. I will switch the screen after 2 to 3 minutes. It's a easy question. Only thing is that you have to pay attention to the two last lines. A student collected all the plants with smooth seeds. What percentage of plants bearing smooth seeds will have yellow seeds? Okay, this is the question. Now try to solve it. One more minute left. Okay, let us try to answer this question now. Hmm. So the question is a cross between two plants that are heterozygous for seed color. So let us try to first understand the seed color. Heterozygous for seed color, green is dominant and yellow is recessive. So this would be GG. Okay. Whereas green is dominant and capital G is dominant, which is green color. Small g is recessive, which is yellow color. And seed coat. Seed coat is smooth is dominant and rough is recessive. So SS. This is smooth and this is rough. So it would be, it's a dihybrid cross, GGSS, GGSS. So what will be the gametes? Gametes would be GS, this, and this. These are the gametes. Okay, it is a dihybrid cross. Now let us make a Punnett square and try to do this. Okay. So we have GS, GS, GS and GS. Okay. Here also this, this, this and this. So now this would be GSS, GGSS, GGSS, GGSS. Okay, here it would be GGSS, 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 GGSS. Then GGSS, 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 GGSS. Okay. Then here GGSS, 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 GGSS. So this is the Punnett square of dihybrid cross. Now they are asking in the question, a student collected all the plants with smooth seeds. Now smooth is dominant and smooth is this one. Seed coat is Z. So this is your dominant one. A student collected all the plants with smooth seeds. Now let us see what all here are smooth seeds. How many smooth seeds are there? Okay. One. Smooth seeds ka genotype is this or this. This is the genotype of smooth seeds. This is smooth seed 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So smooth seeds are total 12. So smooth seeds are total 12. Now in the question they are saying what percentage of plants bearing smooth seeds, means you have to take overall as only smooth seeds, will have yellow seeds. Now green is dominant and yellow is recessive. Okay, seed color mein. So yellow will come only GG. Okay, so GG, small g and small g are yellow because yellow is recessive. So now see amongst this how many yellow are there? 1, 2 and 3. So what would be the probability? What is the probability? The probability would be 3 by 12, which is 1 by 4, which is 25%. 25% is the probability. Because what are they asking? What percentage of plants bearing smooth seeds will have yellow seeds? So what percentage of plants bearing smooth seeds? So amongst the all the plants which are having smooth seeds, what have yellow seeds also? So smooth seeds are total. Smooth seeds were total 12, and out of these 12 smooth seeds, 3 were the plants which had yellow seeds also. So therefore we did 3 by 12, which is 1 by 4, which is 25%. So I hope this is clear to everybody. Now next question is, a mutation on the X chromosome causes a recessively inherited disease. All the children of couple A are affected by the disease. Couple B does not suffer from the disease, while one of the two sons suffers from the disease, the other son does not, and none of the daughters are affected. If the son of couple A marries a daughter of couple B, what is the highest possible chance that their only daughter suffers from the disease? Okay, it's a very interesting question. I think I'll just explain you this question. Okay, let us try to understand the genotype of both the couples. First, we'll try to understand the genotype of couple A, and then for the couple B. Okay, couple A, see, a mutation on the X chromosome causes a recessive inherited disease. A mutation on the X chromosome causes a recessive inherited disease. This means it's a X-linked recessive disease. It is an X-linked recessive disease. All the children's of, now it is an X-linked recessive disease. So it can only be caused when the genotype is X. It can be caused if the genotype of mother, it can only be caused if the genotype is X dash Y or XX dash, X dash X dash. Okay. So it's a recessive disease. So it can be caused if the father has X dash Y and mother has X dash, where X dash refers to the mutated version of X, because it's a X-linked recessive disease. Now all children of couple A are affected. Since all the children of couple A are affected, this means that the father has the genotype of X dash Y and mother has the genotype of X dash X dash. Okay, because all the children are affected. Now let us try to make Punnett square and see X dash Y and X dash X dash. So X dash X dash, X dash Y, X dash X dash, X dash Y. So only when both mother and father are affected, then only all the children can have disease. So this is clear. All children of couple A are affected by the disease. So in order for all the children to be affected by the disease, both mother and the father should be affected by the disease. And what can be the phenotype of father if he is affected by disease? It can be X dash Y, where X dash refers to the mutated form of disease. And since it is an X-linked recessive disease, that means for mother, both the X allele should be mutated, then only it can show the disease. And when we made the Punnett square, we saw that yes, if this is the genotype, all the children are affected. Now we come to couple B. While one of the couple B does not suffer from the disease, that means father is XY, because couple B does not suffer from the disease. While one of the two sons suffers from the disease, the other son does not. That means 50% of the son is suffering from the disease and none of the daughter. That means disease is getting there, but couple is not affected. That means father is normal, but mother is a carrier, because there has to be one mutated X gene also, and it has to be present in such a way that none of the parents are affected. So it is only possible in the case of mother, because father may to, even if there is one, because there is only one X gene, so if it is affected it will automatically show. So this means mother is a carrier. So mother would be X dash Y, and she is a carrier. Carrier means who carries the affected genes but itself is not affected. Now if this is the genotype, let us try to make the Punnett square and see whether it shows what we say or not. X dash Y, X X X X and Y. So here they are saying, while one of their two sons suffers from the disease, yes, one of the two sons suffers from the disease, and none of the daughters are affected. You can see none of the daughters are affected. One is a carrier, other is not even a carrier. So now this is correct. Now they say कि if the son of couple A marries a daughter of couple B. Now you can see son of couple A is disease, which is X dash Y. So son of couple A, son of couple A is diseased. तो उसका genotype is X dash Y. If the son of couple A marries a daughter of couple B. Now daughter of couple B, daughter of couple B, it can be either XY or X dash Y, because they haven't mentioned. So it can be either, sorry, either XX or X dash X. Okay. What is the highest possible chance that their only daughter suffers from the disease? That their only daughter suffers from the disease. What is the chance that their daughter suffers from the disease? Okay. So now there can be two possibilities. First is when the son is marrying a normal daughter, which is not even a carrier. So X dash XY, X dash XY. So here is daughter is getting affected? No. But if a son, the diseased son marries the carrier X dash, so X dash XY, X dash X dash Y. This is the diseased one. So since this is the diseased one, here the chance of getting disease is 50%. Here the chance is 0%. So this overall would be 50%. Okay. Are you able to understand? The question says, what is the highest possible chance that their only daughter suffers from the disease? That daughter suffer from the disease is here. If this is the case, if the daughter has this genotype, then there is 0% chance of the daughter to suffer from the disease. But if the son marries a daughter with a carrier, then there are 50% chance for the daughter to suffer from the disease. So this is the question. It was a good question. Okay, now let us see the last question for the day, which is choose the correct statements. If a heterozygous fly is subject, okay, first I will write A, B, C, D here. A, B, C, D and E. So if a heterozygous fly is subjected to test cross, all F1 progeny will show dominant trait, then the frequency of recombinant phenotype is higher than that of parental phenotype. C. Chances of a viable offsprings are lower if non-disjunction event occurs in meiosis 1 compared to a non-disjunction event in meiosis 3. D. Linked genes are prone to show parental phenotypes in the offspring rather than recombinant phenotypes. And E. Two genes that are 2 centimorgan apart will undergo recombination more readily than genes that are 20 centimorgan apart. Not centimeter, centimorgan apart. So these are the options. Solve this question. Okay. So let us try to solve this question. First option is if a heterozygous fly is subjected to test cross, all the F1 progeny will show the dominant trait. If a heterozygous fly is subjected to test cross, so suppose this is XY, or let us, it is not X linked, let us take it as AA, is subjected to test cross. Test cross is a cross with the homozygote recessive. So it would be AA. Okay. So let us make this cross. So if we make this cross, heterozygous is AA and test cross means this would be eh, this would be AA. Hmm. Hmm. So it would be AA, capital A, small a, small a. So if a heterozygous fly is subjected to test cross, all F1 progeny will show the dominant trait. So is all F1 progeny showing the dominant trait? No. So this would be wrong. So this statement is wrong. Okay. See, I am making it here. This is heterozygous. A, A. And this is the test cross. A. Test cross is with the wild type recessive. So capital A, small a, small a, capital A, small a, small a, small a. So these are the recessive ones. So the question says, if a heterozygous fly is subjected to a test cross, all F1 progeny will show a dominant trait. So this is no. 50% of the progeny will show dominant trait. Okay. The frequency of recombinant phenotypes is higher than that of parental phenotypes? No. Never. The frequency of recombinant phenotypes is never more than that of parental phenotypes. It is always more than the parental phenotypes in the case of linked genes, and even if they are not linked genes, it would be same, but it can never be more than that. Okay. So this is again wrong. Then chances of a viable offsprings are lower Hmm. Hmm. Chances of a viable offsprings are lower if a non-disjunction event occurs in meiosis 1 compared to a non-disjunction event which occurs in meiosis 2. Yes, it is a correct statement because non-disjunction in meiosis 1, it causes all the gametes to be abnormal. Okay. Whereas non-disjunction event in meiosis 2 produces only half of the abnormal genes. Therefore, meiosis 1 non-disjunction generally yields fewer viable phenotypes. So, non-disjunction event in meiosis 1 will generate fewer viable phenotypes. This is correct. Linked genes are prone to show paternal phenotype in the offspring rather than the recombinant phenotypes. Yes. Yes. Why? Because they are linked together. Since they are linked together, so their chances or frequency of undergoing recombination will be reduced because the genes are linked together. So, greater is the distance between the two genes, lesser is the linkage, and there are more chances of recombination. Smaller the distance between the two genes, they are more tightly linked, and lesser are the chances of recombination. So, this is the statement which will help you to solve the last question, which is two genes that are 2 centimorgan apart. This is gene number A, this is gene B, and the distance between them is 2 centimorgan will undergo recombination more readily than genes that are 20 centimorgan apart. C and D, they have a distance of 20 centimorgan. Here the distance is less, here the distance is more. Since the distance between these two genes is lesser, therefore they are more tightly linked. Here linkage is more. Here, since the distance is more, so linkage is less. And since here linkage is more, therefore their recombination is less. And here recombination is more. So you can say that linkage is inversely related to recombination. Okay? Linkage is inversely related to recombination. So, more the linkage, lesser the recombination. And linkage we measure with the help of distance, and that distance is measured in Morgan, basically centimorgan, which we write as. So if the distance between two genes is 2 centimorgans and the distance between other two genes is 20 centimorgans, here distance is less, so linkage is more, recombination is less. Here linkage is less, recombination is more. So this is wrong. Okay. So the correct answers are chances of viable offsprings are lower if a non-disjunction event occurs in meiosis 1 compared to a non-disjunction event in meiosis 2, and linked genes are prone to show parental phenotype in the offspring rather than recombinant phenotypes. So I hope this is clear to you. Okay. So with this, we'll end today's class and we'll meet in the next class, which is on Tuesday.